My name is Christine Curry, and I'm the outreach coordinator of the Iowa Division's Izaak Walton League Watershed Talks. So welcome, everybody. This evening, we have the privilege and opportunity to hear from Professor Claire Hruby and her senior students. We're super excited about that. What do Bulldogs know about water quality? This is our Earth Day edition. Drake University students lead the way in watershed science. And we are going to talk this evening about, oops, um, sorry about
Got it. You go ahead. I'll watch that.
Okay, and we do have Chris Henning here with us this evening, who is co-hosting with us, and she'll be taking care of the chat room and helping us make sure everybody gets in without any problems. So through Dr. Hruby's research program, these students are a part of the soil and water assessment team tracking real-world challenges with applied science. Their work helps identify some sources and impacts of contamination in soil and water systems across Iowa, critical knowledge for protecting both public health and our natural resources. This evening, Professor Hruby will provide a brief overview of the program, followed by short presentations from all of her all-star seniors who will share insights from their individual research projects. So I will let Professor Hruby take it away. Thank you.
Otherwise known as Claire to her students. Well, thanks everybody for coming. It is a beautiful night outside, so thanks for coming inside and watching your screens for an hour here. Um, these four students, Lindsay, Harrison, Paige, and Alex, have been working for a couple years now on projects for the water quality lab. They have all sorts of different funding sources, all sorts of uh, different project areas. And we've definitely hit all of the corners of the state and a lot of Polk County over the last couple of years. But this is my third year at Drake after 20 years at the DNR. And it's really exciting that we've, built on work that previous professors have started. We've started our own projects, and I wanted to make sure you all were invited to our little lab party on uh, on May 8th. So from 1 to 3, we can't bring, well, we can bring snacks in the lab. We're just not gonna serve them in the lab. Um, so we don't, break any rules. But Drake has chosen my lab as one of the projects they put forward to the Carver Foundation for money for improvement in the sciences. And so this is a $175,000 upgrade. Our old lab fit 4 people maybe, and had some pretty fancy vintage equipment. But now we have an ion chromatograph, so we can test nitrate and other ions in the water at with a lot of accuracy. We do E. coli counting. We work on genetics projects, as you'll see, and we have a microscope and just we can accommodate 15 or so people in the lab now, and we're just really excited to share that space and kind of adapt it to whatever students are interested that come along. So you are all welcome on May 8th to come find us down in the basement of Olin Hall at Drake. And with that, I am going to turn it over to Lindsay. Lindsay Siebert just presented last week at the Iowa Groundwater Association, so she is well practiced. She's going to tell you about some groundwater data that she looked at, and then we'll go from there. So I'll let her introduce herself a little bit.
Hello, So my name is Lindsay Sieber. I am a senior at Drake University studying environmental sustainability and resilience. And I've been working with Dr. Hruby for the last year or so on some pesticide and pharmaceutical data that she collected while she was at the Iowa DNR. And in her transition to Drake University, she got those results back. So we started to look at those. Particularly, we focus on when she was doing the testing at the DNR, they focus on vulnerable areas that were alluvial aquifers or areas with shallow bedrock, as well as areas that were in karst areas of Iowa, so the northeastern part of Iowa. Yeah, I think we can go to the next slide. So there were 48 wells that were sampled. You can see the map here showing the locations across Iowa. Majority of them are those red spots there, alluvium aquifers, and then as well as Silurian aquifers in the east areas that are more karst. We wanted to emphasize that this data is of untreated water, so it's not drinking water, but this is still important to look at to understand aquifer conditions as well as for those people who are private well owners. Only 5 to 10% of private well owners test every year because it's not covered under the Safe Drinking Water Act. So there's a possibility that these people are drinking certain pesticides or other contaminants as well. So it's important to take samples like this to see what's going on underground. Also, I should mention Shannon and Dana over at the USGS have been helping us with these results as well, and we're hoping for a publication in the future. Yeah, you can go to the next slide. So here we have the results kind of shown with the detections on the y-axis and the separate wells on the x-axis. So each separate column is a different well with the number of detections of pesticides that were found there. We had 3 wells who were non-detects on the left, and as you go to the right, you see there's a pretty big chunk that had like 1 to 10 detections, and then a small subset of higher detections. The 2 highest detections was that 25 and 18. So
Oops,
yeah.
sorry.
No
You
worries.
should tell me when you want to go to the next slide. I'm just guessing.
Okay, I think we're good to go to the next one. Here we have it broken down with the different kinds of pesticides that were detected. In the green on the left, you see the fungicides, the insecticides in red, and then herbicides in blue. And you can see the majority of the ones that were detected were herbicides. And we also decided to break them down within the parent compound and their degradates to kind of give a better picture of what exactly you're seeing. So a lot of atrazine and atrazine degradates as well as metolachlor. And you can see the parent compound has the asterisks next to the name. So you can see that there as well. I think we can go to the next one. And then here is the breakdown of the pesticide concentrations with the individual pesticide types. These are the top 10 wells out of the 45 that were detected. These ones also happen to have over 1,000 nanograms per liter. A lot of that dark blue there, which is metolachlor SA, and the light blue, which is metolachlor OA. For 66% of the wells, metolachlor degradates were seen as the highest concentration, so that makes sense what we're seeing here, as well as that teal color there, that's the atrazine. And all of these concentrations are under safe or max concentration limits, so they're not inherently dangerous. The atrazine limit is 3,000 nanograms per liter, and what was detected in that second well was 1,700 nanograms per liter. So still under safe drinking levels, but a lot of different mixtures that we don't know much about, like what they're doing to human health or aquifer conditions as well. Yeah. We'll go to the next one. For the pharmaceutical portion of this, there were only 27 wells that had detections and at much lower concentrations, so not as present as pesticides. You can see on the map the size and color composition tells you what pesticide or what pharmaceutical were found where and at what detection. So the smaller the circle, the less that was detected, and the bigger it is, the higher concentrations were detected. You can see there's not really much of a pattern. We do see that blue circles around most of the state. That is N-desmethyldiltiazem, which is a metabolite of diltiazem, which is a calcium channel blocker used for blood pressure medication. You can also see that there's some lidocaine and thibendazole to the east that had some higher concentrations, but overall not much of a pattern there. I think we can go to the next one. And then here we have a box plot showing the variability in the concentrations as well. We have the number of detections on top of each one. On the left, the first three on the left are non-prescription, so caffeine, loratadine, which is your over-the-counter Claritin medication, and nicotine. And context around those ones is a normal 8-ounce cup of coffee is like 90 to 95 million nanograms. One loratadine pill is 10 million nanograms, and a typical cigarette is 10 to 12 million nanograms. And the concentrations that were detected were way lower than that, and only at one detection. And then as you go to the right, you have prescription drugs, which vary a lot more since they're prescribed. But we saw a pretty big variability in lidocaine and thibendazole. But endosmethyldotaism is what was detected the most at 12 times, but it had very little variability, and it was at a very low concentration. So showing that there's pharmaceuticals there, but we also have the breakdown of pharmaceuticals in aquifers. We can go to the next one. So overall, we see that pesticides are much more prevalent than pharmaceuticals and at higher concentrations. And while all concentrations are below drinking water standards, if there are any for the pesticides, the health effects of these mixtures is not well understood and should be addressed for future research or publication. This study only covers a small portion of the vulnerable groundwater in the state. So again, different kind of sampling across the state. I think that's what we got.
If questions pop up, Chris, you're welcome to interrupt, but otherwise we'll just kind of keep going and answer at the end. Okay, so our next presenter is Harrison Ziegler. He's going to tell us about two different projects that he's worked on. And this summer, Harrison is going to go down to Oklahoma and be an intern with the Ogapa Nation and work on some water projects down there near Tar Creek, which is, I think, the largest Superfund site in the country. So that will be interesting. Take it away, Harrison.
Hello, I'm Harrison. So yeah, I've been doing this research in 4 Mile Creek at least for about the past year and some change. And then I've been working in Britton Slough about the same amount of time. I wanted to give a special thanks to Dr. Peter Levi, who started this project off in 2019, Ethan Wing, who picked it back up in 2024, Anthony Seaman, and Polk County Conservation. We can go to the next one. All right. So we've been monitoring from 2019 to 2026. There are some gaps. This data has been passed through many different hands on its long journey to get to me. So there are some gaps within the data set. Weekly grab sampling has been occurring as weather allows. In the winter, we're only able to go about every other week if the water is flowing. So what we do out there is it's grab sampling. I take my sample bucket, triple rinse it to condition it, and then I fill up one of these bottles. It has a sulfuric acid preservative in there to kind of cut off anything that might change the nitrate levels in our sample, right? And then I hold on to these. So our two locations are FMO2 and FMO3. FMO3 is located more near the suburban sprawl in Ankeny, and FMO2 is further out, more rural, though development is starting to push out towards that site. It's mostly agriculture upstream of these two sites. There have been reduction practices implemented, such as prairie strips, saturated buffers, which kind of hold that water and allow plants to uptake a lot of the nutrients out of it before it enters the main waterway, and wetland installation, which wetlands are great. They hold that water, they really slow it down and allow those plants to uptake a lot of the nutrients before it enters the main waterway. So we can probably go on to the next site, or the next slide. All right, here's the graphs. As you can see, there's some breaks in the data where it was past hands. But what we're seeing here is the large graph is the nitrate concentration, the mean of those two sites, each sampling area or each sampling date. And then the smaller graph is the annual cumulative rainfall. So each of those lines correlates to the line on the nitrate graph. They're the same color. And we found that there really it wasn't following the patterns that I had thought it would. So higher rainfall years didn't necessarily yield higher nitrate concentrations. There are some years with lower rainfall, like 2025 has lower rainfall than 2019, but as we're seeing in this graph, has significantly higher nitrate peaks. I'm sure we all remember the watering ban. It was a very high year of nitrate concentrations. We can probably move on to the next. So this is the other project that I'm talking about tonight, Brinton Slough. This is kind of like my Disneyland. I love going out there. It's just a really nice spot, lots of ducks. So Brinton Slough was used as an area that could kind of filter out a lot of the waste coming from a feedlot that closed in April of 2025. So this feedlot had been going for a long time, many decades, I believe. And it just closed in April of 2025. So what we've been monitoring is the different E. coli and coliform levels that are coming in and out of the slough since the closure. And we have data from before the closure. So we're kind of seeing how long it takes for this the E. coli to kind of go down and the levels to kind of return to some sort of a normal. And we plan to study this. I believe that we have at least 5 more years and 10 more years, and we can keep going with this for a long time. So yeah, if you want to go to the next. So these are E. coli levels that we've gotten out of there. So the blue is from the tile and the orange is from the shoreline, so a little bit away from the tile. We found, for the most part, as we expected, the tile is going to be putting out higher E. coli levels than it will be further out as it passes through the waterway a little more. Not always the case, but for most of these data it was. So as you can see, there is a lowering from 2025. It's getting lower and lower, which is good. And we're expecting to see lower E. coli rates as we continue monitoring this area. Yeah, we can probably move on to the next. All right, so my conclusions. They've had efforts to address the nitrate upstream in the ag, but it still remains high. The highest observance doesn't necessarily correlate to the annual precipitation, and it doesn't fit the pattern of the flushing out of the nitrate after the first wet year following a drought. So if it's a drought, it kind of sits there, and then once it becomes a wet year, a lot of that just flows right in, and that's usually the rule of thumb, but it doesn't necessarily fit that pattern. As we've seen, ongoing monitoring needs to be done to evaluate the upstream practices. The changes don't happen, overnight. Obviously, it's going to take a while to see the actual full real effects of these updates that they're doing upstream. And so as we continue monitoring this, we'll continue to get more data and hopefully see lower and lower nitrate levels. For Brinton Slough, it was designed to trap the runoff, working kind of as a wetland to trap, hold it, and lower things before it can go out into the main waterway. I believe it feeds into Beaver Creek. And so, yeah, we plan to continue monitoring that and monitoring the drop in the bacteria levels. And yeah, I'm very excited to see these projects continue once I graduate. I know that they're in safe hands, and it'll be exciting to see the next senior come on here and talk about what they found. Anyways, thank you guys.
Thanks, Harrison. So we're moving on to the northeast part of the state. Paige Robertson is from the Comanche area along the Mississippi. Oh, sorry, my dogs are howling. And And she received some financial assistance from the Darling Institute at Drake, which works to get students out into rural parts of the state. And so she's going to present the work she did last summer at Catfish Creek in Dubuque County, and then some of the results that our team got in the Maquoketa River. Don't forget to tell us about yourself, Paige.
Oh yeah, I'm Paige. It's my third year at Drake. From Eastern Iowa, Clinton, Iowa, Comanche, if you really know. Um, I did work this summer in collaboration with the Dubuque County Watershed. No, Dubuque County Soil and Water Soil. Oh, I forgot what with Dubuque
Yeah,
County.
exactly.
Okay, we can go to the next one. So basically, I tagged along with them during their like monthly monitoring events because they have in Catfish Creek they have um historically high E. coli levels, so they are required to do monthly monitoring. And in addition to this monthly monitoring, I was able to provide them with source tracking, which is basically you're taking a syringe and you're filling it with water, and then you put a filter on the end of the syringe, you push the water through the filter, and then any environmental DNA that's in the water will get caught into the filter. Then we'll put preservative in it and send it off to a lab, and then that lab is able to further analyze what DNA they found in the sample. You can go to the next one. So for our results, I mean, every single one had some human DNA in it, especially TMDLA, which is we can see at the way top right there, that really giant one. That seemed to be kind of the problem area for them. And it, sorry.
We didn't test
So,
for cat DNA. Maybe we should have.
no, she's very adamant. She won't go away. Um, so yeah, all of them had human DNA in them, which is, I mean, a problem. And also some of them did have like cow in them. I think a little bit of dog, raccoon. Um,
Even some white-tailed deer.
yeah.
Yeah.
But the main pattern overall is that there's human DNA in the water. And you can just go to the next one.
Um, yep.
Oh,
There's conclusions in the next one. I think I
oh,
rearranged
okay.
it on you. So yeah. Here, I'll
Is
go
this
to the
for,
conclusions and then we'll go back.
oh, okay. So basically what we found was that there's at TMDL8, there's a high amount of human being detected, which can indicate that there's, sewer problems going on, or maybe there's like there is a Sam's Club near that site just upstream. Maybe the deli is, draining some of their stuff into the water without knowing it's being discharged straight into the creek. But this also was just a tiny snapshot into like what could be going on there. Like if we really want to know what it's like, we would be have to do like consistent data collections to see if we could find any other problems.
Can I go back to Maquoketa? Second there, yeah. So Paige wasn't on this sampling run, but um, The pie charts are similar. We use these for a lot of our projects. Uh, and each watershed we look at, we get kind of different patterns. But again, this was just a snapshot. So, uh, Alex and Harrison, you guys were both on this one. A lot of the tributaries, we saw these little cows peeking in right under the bridge where we were sampling. So not surprisingly, a good portion of the DNA that we found was bovine. And it's probably really small on your screen, but we did sample upstream of backbone as well, kind of right at the north end of the park. And that one was uh, half of the DNA that we found was human, but there was also deer and raccoon and cow and dog in that sample as well. Um, and then down kind of in the Lake Delhi area, on the main waterway where uh, where there are a lot of boats and people fishing, and also a fair number of homes surrounding the lake there. We found a little bit more of the green, which is the human DNA. Sorry, my dog is trying desperately to get into the house in the background here. So yeah, mixtures of sources, a difference between what's coming in on the tributaries and what we find in the more well-used recreation areas. But again, it was just kind of a snapshot to give us an idea and to kind of help the folks in the Maquoketa River WMA kind of figure out what to do next a little bit. So. Paige, the folks in Dubuque, the stormwater folks were gonna look for that pig DNA source at TMDL 8, weren't
Oh,
they?
okay.
I think this one, yeah. So the pink is pig, and that was what, kind of wondering, is it the deli? There's a square in kind of the middle of the suburbs here that might receive manure. We couldn't confirm or deny that. But then I think this was a site you said there were kids playing right in the water there.
Yeah.
So, um, hopefully not too many pathogens, but all of this project is sort of to help inform DNR's TMDL writing. So it'll be interesting to see. I think this is the first project statewide where they have some source tracking along with the regular bacteria and water quality monitoring. Um, Anything else you want to say about that one, Paige?
No. I think to really draw a conclusion, we just need more sampling to be done. Because it was all these were taken within 2 days, I believe.
Yeah. At relatively low flows.
Oh yeah.
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 So that's a common theme in all of these source tracking projects too, and Alex will tell you about that in 2 seconds here. But we want to go back probably to every site at least 4 times to truly do a good job making sure we understand what inputs are happening at what time of year and under what conditions. So we're definitely likely to see a lot more human influence, potentially from wastewater or sewer at low flows, and then more of the livestock or wildlife at higher flows when you have more runoff. Okay, so we will have Alex tell us a little bit more about using these genetic techniques, but this time in Northwest Iowa.
Hi, I'm Alex. I study in the Department of Environmental Science and Sustainability. I am aquatic and earth science track. Thanks to Lakeside Laboratory, I went up there and spent a month up there last summer, thanks to the Ruth Lear Scholarship. I got to go up there and do aquatic ecology, which I highly recommend if anybody wants to go study for like a month or anything, um, just somewhere different. Lakeside Lab is definitely the place to be. Oh, eDNA testing and source tracking in Northwest Iowa. You can go to the next. This right here is Frida Hefner kettle hole, and I believe for me this one was really important because it has a different hydrology. So I tested three different wetlands in Northwest Iowa, and they all have different hydrologies. So I tested a fen, which has groundwater, hydrostatic pressure feeds the hydraulic head. And so I tested a fen, and then this is the kettle hole, which is essentially a pothole from a glacier. Both of those have a lot of peat and okoboji soils. And then I did Marble Lake, which is up towards the Minnesota border. And so that's going to have a lot of the Okoboji soils also, but that's just kind of a pond lake area. So I think that those are really important aspects, especially to the study, to understand just the different assemblages and the different ecosystems within them. So with three different ecosystems, it's really important to understand that when we're looking at these results because none of these results overlap, which is something I feel like is expected because all of these have different pHs and different chemical, like just different parameters all in general. So we did not have any overlap in the DNA processes. So when I went out there, I took all three samples within one day. It was right after it rained, I believe. Um, yeah, it was right after it rained that weekend. I took all three samples to run the eDNA through the filters, which is the same as Paige. They also, oops, sorry, they also run for mitochondrial DNA, which are just small little pieces within DNA assemblage. And so they are able to take those and match them up to different species and stuff. And then they can come back and say, like, oh, this is what you have in here. They can't quantify how many of the species are within the waterway, but they can quantify how many of the fragments they found within the sample, if that makes sense. So kind of vice versa. I wish that we could quantify how many species, but that's, I'm sure, later on down the road. If you want, you can go next. Yeah. All right. So here is kind of just another breakdown of the species that we found. I think it's really cool. We have two different types of biting midges, which I think is really cool. And then we have the non-biting midge. We also have copepods. And through this study, I learned that copepods are predatory. So they actually, like some of the copepods eat different inverts, like within the water column, which is crazy to me because they're so small. So that was really interesting to go through. I also thought it was really interesting that we pretty much sent this in for inverts and we got back some of the different algae, sedges, stuff like that. So I thought that was really interesting as well. I will point out that Marble Lake here in the middle, it only has really one species that came back. And I feel like there needs to be a little bit more quality control study within that because it was a pretty juicy lake at the time. And so I know that there was more than just one species in there. There was a lot of bullfrogs, like how like there was so much going on. So I do feel like we need to slow down a little bit and maybe look at just different DNA structures. You know, do they break down in a certain degree heat? Do they only last X amount of time? DNA acts differently throughout every water column. So, next one.
Yep, these are from the Little Sioux River.
Oh yeah. So these are like from the Little Sioux River. These show in the hydrograph chart the sampling dates so that you can see like where the rainfall was within the water column.
Yeah. So that first sample was November 15th, the very last day of the recreation season. So, we're trying to look at the source tracking, the sources of E. coli. And that was kind of the very last day within the year, but it was a pretty nice day to be out. So,
Awesome.
but in general, we don't worry too much about recreation in late winter anyway. So, and then we went back out in July,
Yeah.
right after the tornado that sent us into a shelter up at Okoboji.
Yeah. That was fun though.
It was. We got to know some other researchers while we sat out the rainstorm. I'm trying to get to the next. There we go.
So these are the samples we got back. I do want to say my eDNA assemblage is different from this source tracking. Same aspect, mitochondrial DNA, just a little bit different. So in these samples we got back, as you can see, a lot of human and bovine. So we have quite a bit of an issue going on there. Um, it could be very well that there's a lot of sewer breaks, old latrines that can be along the way. There's a lot of different aspects that can be contributors to the human DNA issue going on there. Bovine as well. That can be from farmers spreading manure. It can even be from sediment and dirt within the wind. Um, Bovine DNA kind of acts fun. So,
But we did observe, quite a few cattle in the stream, at least in
oh
the
yeah.
upstream portion. And we need to remember that Spencer had floods in the spring, so they're still kind of grappling with the impacts to their wastewater system. So this is where Spencer is.
I then we went back and then we redid the testing. I found it kind of different that the dog was a nice little chunk in the middle there. So, but again, we just have a lot of human and a lot of bovine again. So then we
Yeah,
had a really high, like a chicken sample down here in the
yeah.
south. There was like a
By
few.
Sioux Rapids, there seems to be a little chickens or poultry signature. Uh, we have noticed at other spots, like Darling, where we get the chicken DNA, we have to be careful because people are using chicken liver to fish. And so we're finding some chicken DNA that might be more related to fishing than livestock.
But I think that goes to show how touchy these like test kits are. So like even just somebody fishing, like it's still picking that up and they're still being able to run it, which is nice. So these are the source tracking from the beaches that I did while I was up there as well. I did a lot of source tracking and eDNA assemblage while I was up there. So these are the different results that I got back from the 6 different beaches that I tested. I it's there's a sorry, the coloring's different on here, so I'm like, wait, hold on. The Canadian geese are what was mostly found, right?
Yeah, at least up here at Crandall's Beach. So the blue is
Yep.
is goose but
Yep.
yeah, all of these are really different. And then down at Emerson
Yep.
was goose as
So,
well.
yeah, as we go down, sorry, I couldn't see it because I couldn't enlarge, but now I can. So, yeah, Crandall's Beach, if you've ever been there, it's really nice, but there's a lot of environmental funness that goes on, so it doesn't surprise me about the Canadian geese. The raccoons over at Marble Lake Beach kind of does surprise me because there's not a lot of houses and stuff there, so that one's kind of different. But yeah, there's a lot of them that are just different, and I think a lot of it has to do with where they're positioned at on the lake as well. From like windy days and stuff like that, if there is a high west wind, then it's gonna push. So if you want, you can go to the next. Conclusions. So yeah, source tracking methodology. They are a really simple, low cost way to gather information about different areas. Um, they use a lot of mitochondrial DNA to figure out whatever DNA sources are within the water column. Definitely, additional testing is definitely needed, but not only just to understand the quality and conditions of wetlands, but also to understand quality control of the eDNA test kits as well. So. Thank you.
Sweet. Um, Yay. So thanks everybody. We're hoping a few folks have questions online. There's a whole lot more we could say about all of these projects, but we just wanted to give folks a taste of what we're working on. I agree with Alex that the eDNA for invertebrates has given us some pretty not sketchy results, but just on like surprising results. Our source tracking is really consistent. We always get good data. It kind of makes sense based on what we know about the watersheds. Our very first source tracking was thanks to Chris Henning and the manure spill in Greene County. So we absolutely know that our source tracking method for pig poop works. But ever since that manure spill, we haven't found much pig DNA, which has been kind of one of the bigger surprises. But so we're still kind of curious about using the eDNA for invertebrates, but it has worked really well for source tracking and for fish as well. But that was a different presentation. Okay, I'm going to try to get rid of this pointer so I can see everyone's faces. Let's see. There you Maybe that. There we go. Okay. Sweet. Uh, uh, I see Lee is on here. Lee is up in the upper 4 Mile Creek watershed doing all the good things. I have a feeling that, closer to your farm, Lee, you probably are seeing some pretty significant impacts. There is an open feedlot right above our FM2 site, or more of a pasture area. But there are a fair number of cattle in the stream, so I wouldn't be surprised if our nitrate numbers at 4 Mile are impacted by some other operations between Lee and our testing sites.
Right.
Might be cool to
Right.
add
I
another
have
site up.
two neighbors that have cattle. One neighbor restricts their access to 4 Mile a lot, and the other one doesn't at all. So I think you're probably right, Claire.
We have done one source tracking test. We didn't put it in here, but it came back pretty high for the cattle DNA. So
Right.
definitely playing a role. And if it's really tricky in bigger, when you get into bigger watersheds to see changes in the nitrate, especially with the highly variable signal and the influence of rainfall and other things. So, we're expecting to see improvement given how many folks are working on the upper 4 Mile watershed. And I think, yeah, we need to keep sampling because those long-term data sets, as we know, like from the sensors, are really helpful.
Yeah. I don't think you'll see any llama DNA. I don't let Lenny into the creek. So
Hi, guys. I was just telling someone about your cute llamas, Lee. So if some reporters come knocking and want to get film of your llamas, it's my fault.
Right. So I actually have a question for Harrison, if you have a minute.
Absolutely.
So Harrison, my farm is located upstream from where you're pulling samples. I'm actually on Almond Creek, which is it joins 4 Mile, a mile south of my farm. But I'm curious why you think there might be a chance we'll see less nitrate because I haven't seen any data that shows that. We have a lot of edge of field practices in place, as you know, and Ethan Wing is playing a role in testing saturated buffers and so on my farm. And but I actually haven't seen any data that shows uh, where we improve an individual tiles water, yes, but not creek water. So what What do you think about that?
Yeah, so my hope is that those edge of field practices start to take hold and start to be adopted by a lot more farmers in the watershed, which would hopefully make the nitrate levels go down. So hopefully, as more people adopt those edge of field practices, then we'll see a reduction. I don't know if that answers your question at all.
You don't think there's just enough change in practices to see anything downstream? Is that what you're saying, guys?
Well, I what I know is that my creek, Almond Creek, is entirely tile fed.
That
Uh,
up here,
or
like right where
well,
the word 4 mile is.
yeah, up in that area. So yeah, that's
Yeah.
yeah. And so no spring water that I'm aware of. We have a big county tile coming in actually from Story County. I'm in Polk, but there's a big county tile. It's, I don't know, 24-inch tile or something that comes in. And then 4 Mile actually starts in Boone County, just barely northwest of the Heartland Co-op at Slater. And it's just a ditch. And then and actually goes right by the Slater City water treatment pond. So I'm curious what picks up there. Um, so, but you're right, Harrison. We've put in a lot of edgy field, and some of us are cover cropping. Um, I just, I guess we need more monitoring to see if we, in fact, are making a difference. There's a difference between showing a difference in an individual tile water and in the creek itself. That's what I see.
Thanks, Lee.
And I think it could be interesting to try sampling a little further up the 4 Mile Creek as well, see if that can change or show us any new data. But thank
Right.
you.
Yeah. Thanks.
Yes, you had a question.
I do. Henderson Park, you remember that's one of the first places we met in Greene County.
Yep.
We have a USGS real-time monitor, and the nitrates just hang in between 15 and 18. Later in the spring, I'm sure they'll go to 20 and stay there for several weeks. It's just east around the bend from the sewage plant, which is come online now. And it just struck me that it is also on the bank is the city golf course, not that far from the water. So what do we need to be looking for? Do we need to do a DNA, or do you think we just live with it and know that that's coming downstream to you in 3 days, or what do we do?
So we did some source tracking in the raccoon, thanks to your Raccoon River funding. And the North Raccoon, the sample I remember was equal parts human, cow, and pig, at least in the spring sort of high water sample. And in general, in the North Raccoon, Dan Hogue and others have seen a fair amount of inputs from wastewater at low flows. So Dan has done all the calculations to say how much of the nitrate coming downstream, or the flow even coming downstream at low flows is coming from wastewater. So again, it really kind of depends on the conditions. But Keith Schilling just did a presentation last week at our groundwater conference and talked about, going way back in the past and how we've definitely, made big improvements on the wastewater side. But we have a ways to go. And especially in our sort of headwaters or smaller watersheds, the septic issue keeps coming up. And then here in the Des Moines metro area, we have a lot of leaky like old infrastructure, leaky sewer systems, that seem to be playing a role. Even the creek right next to Drake, which pretty much is, a stormwater outfall that comes out of Drake is got super high E. coli coming out of it, and so there's some leaky sewer systems happening as well. So yeah, but the North Raccoon, the South, raccoon seems to be a lot of cattle DNA, as we know, because there's a lot more feedlots. But the North Raccoon has a chunk of human in there, for sure, at least from the source tracking we've seen. It's more likely to we can say for sure that it's related to sources of bacteria. Um, it's a little hard to source track nitrate because the timing for nitrate is really different than the timing of bacteria transport and how long bacteria survive in the watershed and how long the DNA stays intact in the water. It's a shorter time frame, whereas the nitrate, can sit in the soil for years before it comes out and causes an issue downstream. So I try to make sure folks know that when we're source tracking, it's telling us usually more about the bacteria problems, but can sometimes inform our discussion of nitrate as well. Especially, in class, we looked at some of the nitrate data um, at Van Meter, and there are some times of the year where the increases don't follow the hydrograph in the way that we would expect. So there are some weird peaks in nitrogen, that aren't associated with what looks like a, precipitation event or a tile flow contribution. So we've seen some weird wiggles in those graphs, which tell us that maybe there's a point source contributing. So
Thank you.
sure. Are there other questions in there? Um, Mike likes our invertebrate data. We definitely have a ways to go, Mike, if we're like, and we really like doing the actual macroinvertebrate sampling, right, Alex? So I don't think eDNA is going to replace the macroinvertebrate sampling anytime soon. We get really different kind of results, but we haven't had a project funded where we can do them side by side on purpose more frequently. But DNR, Ken Creer at the DNR worked with the lab that we work with for the genetics and sent them a lot of fin samples. So their library of DNA does a fantastic job when we're trying to identify fish diversity in our streams. And we just kind of, wherever we can find money in the budget, we do eDNA samples for fish because we just want to document what's in our streams. Our fish data in the state are kind of few and far between. But we're not super impressed yet with the macroinvertebrate data. So maybe we need to work with the lab more and send them more samples so we can represent what we have better.
Definitely. I agree with that.
Yeah. Which is
And
really a bummer to poor Ginny over in Polk County Conservation because her volunteers don't really want to do macroinvertebrates, and she was hoping we could just do a little eDNA, but we're not there yet.
But it's so much fun. Okay.
Yeah, exactly. Like we're always going to want to look for bugs. I think that's just how it is. But
So Mike also asks if you want to comment on the Nishnabotna.
yeah, so we did do the fish eDNA for the Nishnabotna. That was April's research, and it's like a year old now. But we did see a recovery of all but one of the species that were on the list from the fish kill in spring of 2024. And our list of species from the eDNA overlapped DNR's list of species from assessments they did after the fish kill. So, The eDNA worked really well and told us the story of what species were in the niche post fish kill. But they don't tell us how big the fish are. And the DNR folks that went out to do the fish stocking and count fish after the fish kill said they had just hundreds of baby catfish stuck in their nets that were, less than 2 inches. So we got, the catfish DNA, but it didn't tell us how big the fish are or, how healthy the population is. So we could use our data to say, the species are back. Probably, a lot of them are coming from upstream and a few from downstream, but we can't say for sure how big those fish are. Um, and that's the extra piece of information which sort of tells us about the health of the populations.
I think that like there's also an aspect in there too that shows that when the leak happened, a lot of the fish were in migration, so that really helped. So there's that
Yeah, they
too.
were still down. They hadn't quite come back from the big river. Um, so if that fish kill had happened a little bit later in the spring, um, those populations might not have recovered quite as quickly. So that's the story that, the DNR folks will also say they were sort of hoping the fish kill killed all the carp. Uh, but it did not. And, the niche, the assemblage of species in the niche is not stunning because the water quality's been bad for a long time. And the hydrology is bad and, all of that. So it's a flashy system with steep banks and a lot of mud. So they were pretty tolerant species to begin with. But doesn't
Mike's
mean we
asking if you could identify the missing species. You said all but one. Do you
it was
remember?
golden eye. I never remember. Um, yeah, it was just that one species. Um, we did. It's interesting because the 3 sites we had on the niche was one above uh Red Oak, one below Red Oak or Redfield. I always get them wrong. Red Oak. Okay.
Red Oak.
Yes. Joni Ernst wearing plastic bags on her feet. That's what I remember about Red Oak. Okay. Uh, but uh, but anyway, yeah, she did, and all the way down at Hamburg. And you could see like the sturgeon DNA in the Hamburg sample and some of the other species that are more likely to be in the big river over winter. So that was kind of cool.
And Mary Ellen's asking, wasn't the niche a straightened river, especially in that area? It is very much straightened, and that's what you were mentioning. Muddy, flashy,
Yeah.
not
Yeah.
not a nice rolling river by
No,
any
no
means.
good riffles, in
No.
that segment at all. So they have to be pretty mud tolerant,
And do
for
I
sure.
remember that there is a lawsuit by the Nishnabotno water defenders, which has come up because of the spill, and they're suing the co-op, aren't they?
They are, yeah.
I don't remember all of the details, but that ought to be an interesting watch here for all of us. Christine, we're coming down the home stretch here. You
Yeah,
want to
we are.
take it back and do a
Sure.
wrap up? I did change in, I hope I changed it right. Your Drake Lab open house is May the 8th. Is that right?
Yes.
Okay.
Friday, May
And
the 8th.
Friday the 8th and 1 till 3 is what I put in the chat. Is that right?
Yep, it's an open house, so you can kind of stop back anytime. If you come right around 1, we'll do a little 5-minute introduction spiel, but otherwise, yeah, just come and you never know, you might even meet my parents,
How
which,
cool is that?
yeah, so,
That's
yeah,
fantastic.
should be fun.
Well, we are super, super grateful to have Professor Claire Hruby here at Drake University and with all of these amazing students. And also super grateful for all of our people who joined us this evening on this beautiful, absolutely gorgeous evening. And so thank you for taking time out to spend a little time with us this evening, and especially the students. I know that this is a busy time for them wrapping up everything. So we are extremely thankful that they took out time to be with us. And what a wonderful opportunity for everybody. I mean, look at all the benefits from this project and all the students' special projects for them and for us and for the future.
Yeah, we have a good crew, and we've already got one environmental science student who's got hired on at DNR writing NPDES permits after graduation. So I feel like we're sending our minions out to take over the world. It'll be good.
That's great. We need them. We need them. All right, everybody, and thank you, Chris, for co-hosting this evening. It's always great to have you and several of the other people on with us this evening as well. So have a
Thanks, Christine.
beautiful week, and this will be up on our website soon, and we'll see you next month with updates. The low
Thanks, everybody.
are scheduled. If all goes as planned. So we'll get to hear a little bit about what they're doing. So thank you again.
Thanks.
Good night, everybody.
Thanks, Christine.